Okay, this is lecture 1 of human physiology, and what we're going to do is cover the following topics. Um, I'm going to start with blood, but then I'm going to cover just the basics, the very simple basics of the nervous system control of some tissues. And then we'll get into cardiovascular, which includes both heart and blood vessels, and then respiratory, muscle, urinary, digestive systems. And then if we do have time, we'll cover the lymphatic system. That's the anatomy, essentially, of the lymphatic and lymphoid system. And then, although I wouldn't guarantee that we'll get there, I'll give you some information on the immune system, which we may not have time for, but we'll see. Okay, so the first topic that we're going to cover is blood, which is a liquid tissue, and it's really the only major liquid, completely liquid tissue, a large volume of liquid tissue in the body. And a lot of these things you may have already covered, but I think there'll be more detail here. By the way, so in terms of physical characteristics, the color of blood varies, and that's because hemoglobin will change in the hemoglobin content of red blood cells changes its color depending upon how much oxygen it's saturated with. Oxygen-rich, fully saturated blood is a very bright scarlet color, a bright red, and it becomes a darker red, even burgundy, if it's less than full, as it becomes more depleted of oxygen. pH of blood is maintained very carefully within a fairly tight range of 7.4 plus or minus 0.05. Temperature of the blood is slightly higher than normal body temperature, which is 37 degrees. It's at 38 degrees. And that's why if you have an area of inflammation, it feels hotter because it's getting more blood supply. And therefore it feels slightly warmer because it's at 38, the blood. On average, blood is about 8% of the body volume, and the volume for males, because males are larger on average, is 4 to 5 liters, and females, 5 to 6 liters, and for females it's 4 to 5 liters, although, you can have significant overlap at times. There are two major parts to the blood. There's the liquid portion, which is called plasma, and then there are membrane-bound elements. Most of them are cells, but platelets are not real cells, and therefore your textbook calls these as elements instead of cells, but really everything's a cell. Everything membrane-bound is a cell, with the exception of platelets. You have red blood cells, or erythrocytes, which are abbreviated RBCs, and you have white blood cells, or leukocytes, which are abbreviated white blood cells, WBCs. And then there are platelets. In order to look at blood, blood is a very accessible tissue. It's very, very easy to get a sample of this type of tissue simply by drawing a blood sample. And that blood sample can then be centrifuged. And if it's centrifuged, then the cells and platelets will sediment to the bottom of the tube due to centrifugal force. And what you'll get once you centrifuge it is plasma, which forms a little bit more usually than 50% of the volume. And although you can't see it, it's not as dramatic as drawn in this picture. You'll see a layer of white cells, both white blood cells or leukocytes and platelets, and that's called the buffy coat. And then the red blood cells or erythrocytes. This is the way it's always presented to students, but I can assure you that in truth there is actually quite a bit of white blood cells in this area here. They're not all in what's called the buffy coat. Nevertheless, there is a buffy coat. It's a very thin layer. It's a little difficult to see, but it is there. So you have those two components of blood. You have the plasma and you have the cellular portion. That's what people usually refer to as the cellular part of the blood component. And that's white blood cells, leukocytes, and red blood cells. There are many different types of white blood cells, and we'll be talking about them. We'll focus, of course, on the major groups that you see in normal peripheral blood. And then there's also platelets. And I wouldn't worry about the numbers so much, but you should realize that you've got a lot of red cells, quite a few platelets, which are very tiny, and then white blood cells. And the red cells, or RBCs, their main job is to transport oxygen. They also are involved in carbon dioxide transport. We'll describe that and talk about it quite a bit. White blood cells are involved in the defensive system or immune system of the body, and platelets are involved in the very first steps leading to blood coagulation. Okay, the plasma portion contains a lot of different components, many different. There are probably at least 100 components, if not more. Mostly dissolved gases and ions and nutrients, many different types of proteins. There are other molecules like hormones. There's waste metabolic products. There's all kinds of things in there. I should point out to you that if the fluid portion is taken, if blood is collected into a tube and there's no anticoagulant, if it's allowed to clot and then spun down, then the fluid portion is not called plasma, but is called instead serum. So serum, in fact, is plasma that has the clotting proteins, and there's a whole series of clotting proteins, depleted. So serum is plasma depleted of clotting proteins, and it can only be isolated from clotted blood. Okay, so if it's not clotted, what's floating around in your cardiovascular system, in your vasculature, is plasma. So as I said, there's more than 100 different components or solutes dissolved in the blood plasma. The major protein in the blood plasma, it forms about 50% of the protein in your blood plasma, and all of these are soluble. And is a fairly large molecule, is called albumin. It's made in the liver, and it's a very important molecule for the control of fluid balance between the different fluid compartments in the body. I want to point out to you that you have two major fluid compartments. There's the intracellular, inside cells, intracellular fluid compartment, And then there's the extracellular fluid compartment outside of cells. And the extracellular fluid compartment is then divided into two other compartments, two further subdivisions. One is the vascular fluid, and the other is the interstitial fluid. That's in between cells, not inside the blood vessel, but in the tissues and in between the cells. And there's always movement between those three compartments. It's continually changing, and it's continually, fluids and molecules are continually moving between those different compartments. So if you have something in your bloodstream, it will move depending on different concentrations and pressure levels. It can move from the vasculature, the blood extracellular fluid compartment, or plasma extracellular fluid compartment, to the interstitial extracellular fluid compartment, and then from there, between the cells, interstitial compartment, into the cells, the intracellular compartment. And we'll talk about that more later when we talk about the vasculature, so don't worry about the terms, but I just wanted to introduce the idea to you. The other kind of major proteins that you have, another major type, are the clotting proteins that I've already mentioned. There's a whole series of different soluble proteins which, when they're activated, will activate each other in a series of steps leading towards a formation of insoluble clot. You also have globulins that contain the antibody molecules that are present in the blood plasma. There are nitrogenous waste products that are not protein. Remember, protein, of course, contains lots of nitrogen. Every amino acid in the protein has a amino group, so it has nitrogen in it. But there are also derivatives from that, non-protein nitrogen-containing substances like urea or creatinine. And then, of course, there are nutrients, glucose, other carbohydrates, amino acids, lactic acid can be found in plasma. There are lots of electrolytes, sodium, potassium, calcium, chloride, bicarb are all important ones, major electrolytes within the blood. And then the dissolved respiratory gases, oxygen and carbon dioxide. And these tables, which you can read for your own information and maybe give you a better, clearer idea. Here it says 60%. I said 50%. Depending on the source, depending upon the book or the person teaching you, there may be some variation. Let's call it 55% plus or minus 5. In fact, if anyone ever asks you what number, to state a number, give it a slight, variation. Here I'm saying 55 plus or minus 5. It'll cover almost all sources, all estimates. So you can see that albumin, which is somewhere between 50 and 60%, is produced in the liver, as I said earlier, and has an important role in maintaining fluid balance between the different fluid compartments in the body. Let's move on. Okay, as I said, if you take whole blood, well, of course, when you take a blood sample, you're taking whole blood. You can spin it down, and then you have here in this area, these are called packed cells because they're separated away from the fluid plasma. And they form the majority of the cellular component of the blood. So the cellular component, or better said, I suppose, or more precisely called formed elements, because there are also non-cellular platelets. In terms of numbers, there are the most numerous component are red blood cells, erythrocytes, and then platelets, and then white blood cells. And here's a breakdown of the white blood cell compartment. The most numerous type of, well, I should tell you that these three types of cells are called granulocytes. They're characterized by having granules within their cytoplasm. There are three different types: neutrophils, eosinophils, and basophils. And these neutrophils are the most are in the highest percentage of any of the white cells in the body. They're not only characterized by the fact that they have granules in their cytoplasm, but they also have a very odd-shaped nucleus. It's multi-lobed, and each lobe is connected to each other by a thin strand of nuclear material. So sometimes you look at it under the microscope and you see what looks like multiple nuclei, but it's only one. You could see 2, 3, even 4 separate lobes, but they're each connected by a thin strand. So the neutrophils, the eosinophils, and basophils are all called granulocytes because there are granules in the cytoplasm. Another major kind of white blood cell are the monocytes. Sorry, I'll do that like this. Monocytes, and also lymphocytes. Now, your textbook calls these two populations agranulocytes. Don't use that term. Nobody uses this term agranulocytes. It's never used. It's used by your book, but that's about it. Okay. There's even an entry in Wikipedia that says that. It's an inaccurate term, particularly because granulocytes and monocytes have a common lineage in terms of the cells that they differentiate from or come from. So it's a mistake to lump these monocytes and lymphocytes together. So the three major types of cells are granulocytes, monocytes, and lymphocytes. And the granulocytes have three different types of granulocytes: neutrophils, eosinophils, and basophils. And I'll get into some of the function of these in a little while. So these three are the major cells or formed elements. The white blood cells are in fact complete cells because they have a nucleus. Red blood cells do not have a nucleus. They start off with a nucleus when they're being produced in the bone marrow, but then they eject the nucleus. In the end, they really are just simply, for the most part, a membrane-bound bag of, or unit of hemoglobin that's carried around, that has a role in gas transport, oxygen, carbon dioxide, solubilized oxygen, carbon dioxide transport. The platelets are parts of cells. In your bone marrow, where all blood cells originate and come from, there's a type of cell called a megakaryocyte. I'm going to write it here if I can. Megakaryocyte. And platelets are actually fragments that bud off of these and get released. So megakaryocytes, mega because they're very large types of cells, are found in the bone marrow, and the platelets bud off of the surface and get released into the peripheral blood. That means the blood going through your cardiovascular system. Megakaryocytes. It's a little difficult spelling with this, but now, most form elements survive in the bloodstream for either days or weeks, although some can live for a very, very, very long time, years. But most of them only for a reasonably short period of time, and therefore your cells, blood cells, are constantly aging and dying off. They get aged, and then the aged or older cells are taken up by cells in the liver typically, and the components are recycled. And new ones are constantly being formed in the bone marrow and released into the peripheral blood. The red cells do not divide in the blood. They come from the bone marrow, and all the white blood cells are also produced in the bone marrow. And some white blood cells, if they're activated, can divide in the blood, however. Okay, so here what you see in this photomicrograph, in other words, a photograph taken with a microscope, is a smear of peripheral blood, a sample of blood taken from the blood vessels. So this is taken from a vein, like most blood samples are taken from the vein, and then it's smeared across. A thin smear is made on a glass slide, and then it's stained with a very routine stain. And what that does is it will highlight the nucleus. And remember, all of these other cells all around here, all through this area, everywhere, the most numerous cells are red blood cells, which do not have a nucleus. The cells with containing a nucleus are white blood cells, and then here in this area here, what you have are some clumped platelets. It's very difficult to not get. As soon as you draw the blood, they start to clump together. So it's very difficult. There are many platelets here. They're very small and they're all lumped together. Now, if we look at the individual cells, these are the red blood cells. Red blood cells have a unique kind of biconcave disc shape. They edge on, they look like this. It is a biconcave disc. It has essentially, not the greatest artist, but that's what it looks like on edge. And here is from the top down. So here's an example from the top down. I don't see any examples on edge, but if you did see it on edge, it would look like this one here. It has a high surface area to internal volume ratio, which means you can get an increased interaction with the plasma around it, which is good for exchange of gas, dissolved gases. The reason why these cells in the photomicrograph have a clear center is because it's thinner in the middle, and so the light shines through more. Sometimes it can actually be quite bright in the center because it's almost acting like a lens under the microscope, focusing the light upwards. Okay, so those are the red cells, erythrocytes. And here you see a diagram showing that edge on or top view, biconcave disc without a nucleus, so these are anuclear cells. They do have some internal membrane-bound organelles, but not a lot, mostly filled with hemoglobin, a soluble protein, which is involved in binding oxygen and can also bind some carbon dioxide. The membrane itself contains some proteins that give it quite a lot of flexibility, because these red cells have to travel through capillaries and often will bend as they go through. This is a good example of the biological principle of complementarity between structure and function, that a particular biological component in an organism has a certain function because of its structure. The structural characteristics here contribute to its ability to take up and release gases and transport it, large surface area relative to the internal volume. One fact that a lot of people actually aren't often aware of is that in terms of ATP production or energy production in the cell, all of it in red blood cells, unlike other cells, is generated by anaerobic processes through anaerobic fermentation or lactic acid fermentation. In that way, they don't consume any of the oxygen they're transporting. It would be conflict of, I suppose you could say, if it had a consciousness, a conflict of interest or a conflict of purpose if it was carrying out aerobic respiration to generate ATP. So they only carry out anaerobic respiration or fermentation. They're essentially dedicated. They're a bag to respiratory gas transport. As I said, it's a bag of hemoglobin. And hemoglobin, often you'll see hemoglobin abbreviated as HB or HGB like this. It has an unusual characteristic in that its job is to not only bind, but also release oxygen. So it will reversibly bind oxygen. Almost all of the blood, in fact, almost all the oxygen in your blood is, in fact, bound to hemoglobin. This is a diagram, an artist's diagram of hemoglobin molecule. It's not really a classic kind of diagram, but it's showing that there are 4 polypeptide units or subunits. Actually, it's calling it a chain, but by definition, strictly, proteins that contain multiple strands of protein that are linked together by disulfide bonds are called chains, and if they're linked together by non-covalent bonds, disulfide bond, of course, is a covalent bond. If they're linked together, if the polypeptide strands in a multi-stranded protein are connected to each other by non-covalent bonding, like hydrogen bonding, then they're called subunits. So strictly speaking, it should be said that these are 4 subunits rather than 4 chains, but a lot of people will use the terms, including me, will use the terms loosely. Chains, they call everything a chain. So here you have 2 identical alpha chains shown here, and 2 identical beta chains. The protein is globin, and at the center of each globin chain or subunit is a heme group, and this it shows the heme group here as a flat circular disc-shaped structure, and here's a blow-up of it. And at the center of each heme group is an iron, and it's the iron atom which can bind to one O2 molecule, one molecule of oxygen. And therefore, because you have 4 hemes within each of the 4 polypeptide subunits of hemoglobin, you have, when fully saturated with oxygen, it can transport 4 molecules of total. Fully saturated, it will carry 4 molecules of oxygen. Normally, and it varies. These numbers vary again depending on the source. You might see slightly different numbers, but there is overlap in the 2 numbers. Men typically have a higher here. We haven't talked about this hematocrit yet, so I won't highlight that right now. So hemoglobin typically is higher in males than in females, and that's in the blood, and that's because the percentage of red blood cells within the blood of males is higher than in females. The hematocrit refers to the percentage of red cells within the total volume of the blood. So if you took a tube let's draw it like this. If you take a tube and a sample of blood and you spin it down, and so you end up with, you end up with the red cells down here in this tube, say like that, and the plasma above it, or if it was clotted, it's the serum above it. Then typically, on average, in a male, you would get about, if you looked at the percent, what percent does this volume, what percent is that volume of the total volume? That's the total volume, right? Then you would find that it's typically in a male, that percentage, which is the hematocrit, is approximately 47% plus or minus 5. And in a female is approximately 42% plus or minus 5, right? And again, depending on the source, you might see slightly different numbers, but essentially within this ballpark figure. That's the normal hematocrit, male versus female. There are 3 reasons, actually, why males have higher hematocrits than females. One is that on average, the body mass of males is bigger. The mass of muscle tissue is larger, and that has to be supported by delivery of more oxygen. So one reason is muscle mass. The second reason is that females menstruate monthly. An adult female will menstruate, lose some blood each month. And the third is that males have more testosterone, produce more testosterone than females. And testosterone is a driver of what's called hematopoiesis, erythropoiesis actually. Erythropoiesis, and we'll get to that term later, but that's the production of red cells. So with more testosterone, there's more red cell production. So those are the 3 reasons why males have a higher hematocrit. Muscle mass, increased muscle mass, menstruation in females, and higher testosterone in males than females. Okay. Now, strictly speaking, although I must say you don't hear it that often, but strictly speaking, hemoglobin, if it's bound with oxygen, is called oxyhemoglobin, and without oxygen is called deoxyhemoglobin, and that's probably the last time you'll hear me say those terms. Hemoglobin can also bind CO2, and that's when it's bound with CO2, that's called carbaminohemoglobin. Just letting you know. Okay, the production of blood cells, all of the blood cells, any blood type of blood cell, occurs in the bone marrow, and it is a process called hematopoiesis. Hematopoiesis. And hematopoiesis is going to occur in red bone marrow, not the inside bones, many bones. It's not red bone marrow, it is yellow bone marrow, it is mostly fat. But in certain bones in the body, particularly bones not in the limbs, but the central core of the body, which is called the axial skeleton, that's where most of the red bone marrow is. The ends, the growing ends, or the ends rather, of the humerus and the femur do have some red bone marrow. But most of the richest, I'd say, source of bone marrow would be the sternum in your chest, and also the part of the pelvis, the iliac crest of the pelvis. The cells that produce the different types of blood cells, the stem cell, the most primitive type of stem cell that can lead to or give rise to all the different types of different formed elements or cells present in the bone marrow and then produced there and released to the peripheral blood. That stem cell, that primitive stem cell, is called a hemocytoblast. And so you have hemocytoblasts, and in terms of red cell production, which is called erythropoiesis, remember, production of all the different types of cells is called hematopoiesis, but just for the red blood cells, it's called erythropoiesis, and that goes through a series of steps where you have division, so you have proliferation of the cells, and differentiation. They change as you get that, as you go through the steps, the different phases or types of cells or stages of cells to get to a mature red cell or erythrocyte. Then at each point here, you have both multiplication, proliferation of the cells, and you have differentiation. So you're getting more and more cells as you go this way, dividing and changing, and they go through all these different stages till they get to the mature erythrocyte. I don't need you to know all of these different phases of the differentiation process or development towards a red cell. I just would like you to know the primitive type of stem cell, the hemocytoblast, and the mature cell erythrocyte in this erythroid lineage. Okay, this is called an erythroid lineage. The line of different types of cells leading towards red blood cells, mature red blood cells in the production process, production differentiation process. This is the erythroid lineage of cells. Not that easy to write. There we go. So you have a hemocytoblast transforming into a committed cell, the first one, and it's committed because once it goes in that direction to that form, it will lead to red cells. It's not going to lead to another type of blood cell. It's committed towards that lineage. So they go through different stages. This goes to this. I don't need you to know these terms. I just would like you to know that you're going from a hemocytoblast through different differentiation phases in the bone marrow till you get to a mature red blood cell or erythrocyte, and that's what gets released into the peripheral blood, the blood vasculature. Just before they're released, the nucleus is ejected. Then it goes to this phase. I wouldn't worry. I don't need you to know it. And then those cells, which happen to be called reticulocytes, become mature red cells and leave and go into the blood vasculature. The process, this whole process, is outlining the different stages in the erythroid lineage, showing the different steps in the production of red cells, which is called erythropoiesis. So how is that regulated? Well, the body tries to keep a steady amount of red blood cells, although that can change with certain stimuli. It really tries under normal conditions. It's relatively constant and reflects a balance between production of new red cells and destruction of the aging or older red cells. They don't have an infinite lifetime. If you don't have enough red cells, that can lead to less than normal, suboptimal level of oxygen in the body tissues, and that's called tissue hypoxia. Hypoxia is abnormally low oxygen. If you have too many red cells, then you can get if the hematocrit is higher because the proportion of red cells is higher than normal, then that can lead to a thickening of the blood, so the viscosity of the blood becomes higher than normal, and it's not a desirable situation. Overall, the production of red cells is controlled by a hormone, and I'll tell you what hormone it is later. And of course, you need adequate supplies of all the components that go into making a red cell, including iron, which is at the core of each heme molecule, and also the amino acids that go towards making the globin portion, the protein portion, four protein strands or subunits. Also, B vitamins are required for normal, adequate red blood cell production, or erythropoiesis. Red cells, as you know, transport respiratory gases, oxygen, and they're also involved in transporting carbon dioxide. They're constantly being produced in the red cells, in the bone marrow, the red bone marrow. The hormone that's involved, which will stimulate red blood cell production, in other words, cause hemocytoblasts to start differentiating and producing cells along the erythroid lineage. It is a protein hormone called erythropoietin, and it is produced in the kidney. So it's a renal hormone. And characteristically, this hormone, erythropoietin, is produced in response to tissue hypoxia. If the kidneys are not receiving adequate oxygen, they will start producing and releasing this hormone into the bloodstream. All hormones are released into the bloodstream, and they will travel around through the body until they get to whatever tissue or cells have a receptor for erythropoietin. Remember that signals, in a multicellular organism, you have specialized cells with different jobs. Okay? In you're a multicellular organism. You have lung tissue, brain tissue, blood tissue, fat tissue, muscle tissue, all kinds of different tissues that are specialized. And they send signals to each other. And, the signal as hormone signals are released in the bloodstream and travel around through the whole body, through the blood. But they'll only have an effect on cells that have the right receptor. Receptors are always very specific, typically specific. So if a cell has a erythropoietin receptor, it will bind erythropoietin. And the hemocytoblasts in your bone marrow do have erythropoietin receptors, and this will trigger them to be committed to start producing new cells along the erythroid lineage. So the new cells they produce will be committed towards red blood cell production. And it's always in response to hypoxia, low oxygen. Now, low oxygen could be triggered in a number of ways. It could be because you have a lower hematocrit, a decrease in the red blood cells, or the available oxygen is lower. If you are if you're a normal person, then the only way that availability of oxygen is reduced is if you go to a higher altitude, and that certainly will trigger erythropoietin production. If a person moves to a higher altitude where the PO2 is lower, the partial pressure of oxygen is lower, then that can cause a hypoxia, which the kidneys detect, and they start to produce erythropoietin, and you'll produce more red blood cells. This, of course, is why some athletes will train before major events. A few months before major events, they will train at a high altitude location, so that their red blood cell proportion of red cells, their hematocrit, will actually rise. Another way for tissue hypoxia to occur is if the demand for oxygen is increased. And in fact, the overall tissue demand, for example, in males is higher for oxygen than females because of the increased muscle mass and often use of that muscle. And so they will have a somewhat higher erythropoietin and therefore higher red cell production. So if you have increased production of red cells, increased erythropoiesis, your red blood cell count will go up and oxygen carrying capacity of the blood will go up. And therefore, if you have more oxygen, you can generate energy through aerobic respiration, ATP production of energy in the form of ATP. You can generate more ATP, which is why, again, of course, that athletes want to have a higher hematocrit. That way they'll have a higher hemoglobin level and more oxygen carrying capacity and can deliver more oxygen to their tissues. Well, overall, the mechanism of regulation of blood cells involves this key protein. So the mechanism of regulation of erythropoietin and its production all depends upon the normal blood oxygen level. It's a question of maintaining a normal blood oxygen level that's adequate for tissue oxygenation, and that's regulated by the kidneys detecting the level of oxygen available and responding if it's too low by producing and releasing erythropoietin. So if it drops, if there's an imbalance and you have hypoxia, okay, because you have a lower amount of red blood cells, or you have less available oxygen, or you have a higher demand in your tissues for oxygen, then you will get production of erythropoietin from the kidney, and that will affect the red bone marrow and trigger hemocytoblasts to produce more red blood cells. And that enhanced production of red cells will cause a rise in red blood cell count and therefore a higher oxygen carrying capacity, and therefore the blood oxygen level will go back to the normal level or adequate for your needs. Overall lifespan of red cells is anywhere from 100 to 120 days, and as they become older, aged, they become more rigid and more fragile, and the hemoglobin, proteins don't last forever in the body. They break down, they begin to degenerate, so the hemoglobin inside begins to degenerate. And that change, at least the change in the surface membrane, there are some characteristic changes in the phospholipid bilayer of the red cells, can be detected by cells in the liver. The cells that detect them, those changes are macrophages and some liver macrophages, which are called Kupffer cells. If you didn't know, liver macrophages are called Kupffer cells. These are macrophages that are normally present or resident within the liver, Kupffer cells. And they have many jobs. So one of them is to detect aged red blood cells and phagocytose them and break down the components. The heme and globin of the hemoglobin, the two parts of the hemoglobin molecule are separated, and the globin is, of course, is the protein strands, the 4 protein globin polypeptide strands, and they get broken down to their individual amino acids, which can then get used by the cell or get the macrophage, or released and used by some other cell. The iron is typically salvaged for reuse. Iron, the body tends to never lose any iron that it takes in. If it takes iron in, it tends to hold on to it very tightly. So any iron salvage from red blood cell aged red blood cell breakdown is recycled for used for new red cells, new hemoglobin production. So you have a constant turnover of the red cells. The heme part of the hemoglobin goes through quite a complex series of conversions to different molecules. One pathway of that leads to the production of a molecule called bilirubin that occurs in the liver, and the bilirubin gets secreted by the liver into the intestine in the form of bile. So bile contains bilirubin. The liver produces bile, and it contains bilirubin, and that gets secreted into the duodenum, actually, in the small intestine. I should say small intestine here. As I said earlier, the globin protein part of the hemoglobin, these polypeptide chains, subunits, what have you, get broken down into amino acids that can be reused, released to the circulation, reused by other cells. This is a diagram that goes through the different stages of red cell production and release and breakdown. And I think this is a good place to stop, and we'll start up the next video with this slide. So that's slide 32 of 59.